drugs, and what is the ANDA approval process, and the Indian generics companies involved and how they are performing, and used by FDA. So to begin with, what are the generic drugs?
Sir, Rammohan sir, sorry for interrupting. Net issues.
Can you Yeah, it got disconnected.
Yes, sir.
Can you see my screen?
Yes, sir. Now it is visible, sir. I can see.
Hello, can you see my screen?
Sir, your screen is not shared, sir.
Dr. Munir, can you see my screen?
No, sir.
Okay, so shall I continue now? It got this.
Sir, please try to share your screen, sir. Sir, we request you to share your screen, sir.
Can you see now?
Yes, sir. Now we can see, sir.
Okay. Sorry about that. What are the generic drugs? So generic drugs, it is a medicine which works in the same way as a branded drug. So in the United States, generic drugs are approved in the same way as branded drugs, and they constitute about 90% of the total prescriptions filled. And but still, it accounts for total 20% of the prescription drug spending. So that means it saves a lot of money for the public, but still, they work same as a branded drug. So what are the generic drugs which should have approved by FDA? So it contains, and it should contain, a ingredient which is same as branded drugs. Okay, so there should not be any difference in the salt forms or any difference in any other forms, but the active ingredient should be same as the branded drug. And the generic drug has the same strength and dosage form and the route of administration. So it should not differ from the branded drug's strength, then it may not become a generic. So if the strength is different and the dosage form is different from the branded drug, then it cannot be called as a generic drug. And the
Uh, Ram Mohan sir,
drugs are manufactured the same way, following the 21 CFR 210 to 11
Ram Mohan sir,
standards,
sorry for
which are
interrupting.
called current good manufacturing practice. Yes.
Sir, your voice is not audible, sir. It's not clear.
Yes. Please continue.
Sir, no, sir. It's not clear, not audible.
Hello.
Sir,
Is it clear now?
sir, it is clear, sir. Sir, we request you to kindly hold the mic.
Can you hear me now? How about now?
Yes, sir. Now it is okay, sir.
Okay, I think there is a problem in the internet. Okay, it's not because of my mic.
Yes, yes.
Shall I continue now?
Sir, please, sir. You continue, sir.
Okay, sorry about that. So we were discussing about the generic drugs. What are the requirements for a generic drug to be approved? So as we discussed, the molecule must be same as the branded drug, and the dosage form, strength, and also the generic drug must be bioequivalent to the brand. So generally, when we discuss about the, we discuss, it is approved by a regulatory agency in any country, same as in India. But who regulates these generic drugs in the United States? So there is a regulatory body called the Food and Drug Administration, which regulates branded drug as well as generic drug. So let's discuss briefly about the history of drug regulations in the United States. In 1906, there was a
Excuse me,
Pure
sir.
Food
Rammohan,
and
sir.
Drug Act, which, yes,
Sorry for interrupting. Sir, your screen was not shared.
yes,
Sir, we request you to share your screens.
yeah, I already shared.
Yes, sir.
It is
Now,
shared.
now it is visible. Sir, Sir, please continue. Sir.
Okay. So, yeah, I was talking about the regulatory body which approves the generic drugs. FDA is part of the Health and Human Services, which is led by the commissioner, and commissioner reports to the HHS secretary. HHS secretary is same as the health minister in India. So, let's discuss about the brief history of how the drugs are regulated in the United States. In 1906, there was an act, Pure Food and Drug Act, which prohibited the interstate commerce, and the interstate commerce prevented adulterated and misbranded drugs commuting or transshipping across the straits. So the law did not require drug manufacturers to demonstrate safety or effectiveness, but it was only prohibited the adulterated and misbranded drugs. In 1938, the Federal Food, Drug and Cosmetic Act was enacted, which is now called the FD&C Act, which requires that the drugs be proven safe before manufacturers may sell them in the interstate commerce. So after the enactment of the FD&C Act, all the drug manufacturers needed to prove that the products which they manufacture are safe before they commercialize the product into the market. So FDA reviews the safety data, and FDA approves. Upon approval, the manufacturers can introduce their product into the market. In 1962, there were some deaths because of the thalidomide, and everyone knows about the seal babies. Thalidomide resulted into seal babies, and because of these seal babies, the product was banned that time. Then the new act came into existence. The act was called Kefauver-Harris Drug Amendments. So these amendments brought in the safety provisions also. So in addition to the efficacy, the amendments brought safety provisions, and that requires the manufacturer should show their drugs must be safe as well as effective before the product is entered into the market. In 1984, drug price competition and patent term restoration act of 1984. So this act comprises the balancing greater patent Hello. Hello.
Sir,
Can, can you can you see my screen?
yes, sir. Now we can.
Okay. So after the Hatch-Waxman Act, the generic drugs, whoever introduces the generic drug, first generic drug into the market, they get 180-day exclusivity as a incentive for bringing the generic drug into the market first time. So we should discuss about the different types of the different types of the applications which requires FDA approval before marketing the product into the United States. So basically there are two types. One is the new drug application, which is governed by the FD&C Act 505B1 or 505B2. And the second type is the abbreviated new drug application, ANDA, which is governed by FD&C Act 505J2. So all the generic drugs are approved through abbreviated new drug application process. So the new drug application, when the sponsor of a new drug believes that enough evidence on the drug's safety and effectiveness has been obtained to meet the FDA requirements, and through the extensive studies conducted through clinical studies and also non-clinical studies through the IND route, and then generating the chemistry manufacturing controls data, the sponsor submits that data to FDA as a new drug application. So the new drug application data includes basically clinical studies and the animal, any toxicological or preclinical studies, and the manufacturing stability data and the impurity profiles, method validations, and the biopharmaceutics and the statistics. And ANDS contains basically, after the Hatch-Waxman Act, okay. So the ANDAs, they demonstrate that the generic product is same as the branded drug. When we talk about the branded drugs are also called the reference listed drugs, upon which the generic drugs are compared for the bioequivalency as well as for the pharmaceutical equivalency. So the ANDA, rather than replicate and submit the data from animal clinical and the bioavailability studies, generic drugs relies on the previous findings of the safety and effectiveness of the branded drug. That's why they are called abbreviated new drug applications. So ANDA holders doesn't need to demonstrate the clinical and animal studies. and they should provide that, that the pharmaceutical equivalency is established by way of proving that, the active ingredient is same, as well as the dosage form and the strengths are same, and then the dissolution profiles matches with the reference listed drug, and also the bioavailability studies are conducted. It's called bioequivalency studies. So to obtain the pre-market approval of any generic drug, the sponsor submits the ANDA, which includes basically, the chemistry data as well as the bioequivalency data. Okay. So the generic and branded drugs, they basically differ in certain characteristics within acceptable range of acceptance. That means, they could differ with excipients or a different packaging material, or sometimes, different shape, with acceptable range of differences. Okay. And hello. Hello.
Sir, please.
Okay. So, who, how is it handled in the FDA? FDA has a office called Office of Generic Drugs, OGD, within the CEDAR, the Center for Drug Evaluation and Research, which ensures the high quality and affordable generic drugs are available to the American public. So the OGD is the main office which handles all the generic applications. OGD review process includes managing the
Sir, we
regulatory
request you to
process.
share screen. Sir, sorry for interruption. Sir, kindly request to share the screen.
Can you see now?
No, sir.
Yes?
Not yet, sir.
Can you see the screen now?
Sir, now we can see, sir.
Okay, good. So OGD review process includes managing the regulatory process to facilitate drug approvals and establishing science initiatives, like, developing the new guidances for products and product specific bioequivalency guidances, and publishing the data and reports on generic drug developments, such as, the number of approvals, and how the review process is continuing within the office each month, what are the statistics. So they publish all the data, and also they offer educational materials such as webinars and white papers, and lot of seminars, workshops. Okay. Office of Generic Drugs consists of different offices, and OGD is headed by Dr. Sally Chow. She's the director, and there are other key offices within the Office of Generic Drugs which are involved in the approval process of generic drugs, and they either review or inspect the sites, or both. Some of the offices do both operations. So the FDA's drug program consists of, basically the OGD functions as a key office for all the applications, receiving, triaging, and then collecting all the comments from the different departments, and then communicating with the industry. And provided all the review assessment is acceptable, then they issue approval letters to the industry. So some of these offices, which are under the CEDAR guidance, but they work independent of OGD, and they interact in each individual cases, uh, in collective, to get the approvals. So the total current approvals, if we take the statistics from the FDA website, 2021 has received 776 approvals, which includes total final approvals as well as tentative approvals. So the ANDA process consists of two approval processes. The final approval process includes everything. The final approval letter, which after receiving the final approval, any company can market their product into the United States. And the second approval is the tentative approval, which is not the final approval, but the tentative approval consists all the chemistry data, and the bioequivalency data is reviewed, and even the facilities were inspected, or at least assessed that they are following the GMPs. But the agency cannot give the final approval because there must be some patents still existing, or a patent or exclusivity is still valid. So until unless the patent or exclusivities expire, FDA cannot give the final approval. So they are called tentatively approved. So if we look into the statistics from past 6 to 7 years, the number of ANDAs, either final approvals or tentative approvals, reduced drastically this year, previous year. So the reason could be multiple. This is a bar graph which shows clearly the statistics starting from the January 2021 to December. They are reducing, and if we discuss about the 2021 approvals, there were total 93 first generics. That means the first time they got approval after the expiry of the patent. Okay, so either they were approved after the patent litigation, or the expiry of the patents. Okay. So which is really a good number. From it was 72, and in 2021 it became 93 first generics. And some of those first generics are listed here. Very good products. They were first generics, and based on these first generics, US public have saved a lot of money. Usually, the generic will cut the cost of branded drug up to 1/10 and sometimes even 1/20th time, of the branded drug. So FDA also approved another process called competitive generic therapy. So competitive generic therapy includes, any drug which has very less competition in the United States. Those are called, competitive generic therapy. At least, not more than one approval available in the Orange Book, which is the, publishing list of ANDAs or NDAs approvals. So the competitive generic therapy provides a 180-day exclusivity for because of the inadequate competition, generic competition. And the program was first introduced in 2017. FDA issued that final guidance in March 2020. Okay. And so far, 119 generic drugs were approved through the competitive generic therapy process, and 2021 has seen 52 approvals. And because of the COVID-19 effect, there were some reduction in the NDA approvals also. And in 2020 and 21, there were site inspections also reduced on-site inspections because of the COVID-19 infection. And Director Sally Cho says that, even there were unique challenges, but still UGD continued to, innovate and conduct scientific research to keep the generic drug program moving forward. So they approved 75 ANDAs, which are purely for the COVID-19 related supportive therapies. So coming to the Indian generic companies, how they fared during these, past 4 5 years, and last year 2021, with respect to the other generic players. So the total Indian pharma major companies, they secured 210 ANDA approvals, which is like 30th number, and we really should feel proud. Most of the generic companies which supplies the generic drugs into the United States are located geographically in India. Okay. And in year 2020, they received total 276 approvals. They are like 36.3% of the total. And the total tentative approvals declined in year 2021 to 116 as against 125 in 2020. Yeah, because of the COVID, and also, the number of ANDA applications reduced. Because of that, the total approvals also reduced. So there's a brief statistics about how Indian companies secured the approvals, each company wise, and this is a small list, which is very good, because the total approvals consist 33% of the total ANDAs approved by the FDA. And there was a discussion on the why there is a decline in approvals in year 2021. The total decline could be, because of the COVID, or because of the total number of 1st cycle approvals were flat. Okay, so If we see the numbers are totally, lowest since 2016. And this is likely due to decline in submissions, and submissions fell the fourth year in a row with only 808 generic applications sent to the agency. And we know the conventional review process. FDA reviews the applications, electronically submitted through FDA submission gateway, and if the ANDA is in line with the FDA guidances and all the data is acceptable to FDA, then they will approve in the first cycle, or they will send a complete response letter or information request letters. And then upon receiving industry response to the letter, and then either in first cycle or second cycle, FDA will approve. FDA also thinking of introducing a new process for future reviews. That process is called knowledge aided assessment and structured application, CASA, and modernize the total quality assessments of the applications. Instead of receiving the raw data, or the paper form of the data, FDA requires the industry to submit the data in a more structured manner. And the CASA system captures all the information, and then they will analyze and they will assess the complete data as per the statistics. Also will help with the risk controls. They are related to the product design or the product manufacturing, or during the stability studies or other process. FDA moves system, which is a text-based current assessment system, into a complete unit, issue-based regulatory and technical assessment using the structured data. Hello. Hello.
Sir, sir, kindly continue.
The new review process, CASA. So this is the roadmap for the Hello. Hello.
Sir,
Okay. I- so this is the CASA roadmap FDA has designed. The CASA FDA started in 2015, and in 2021 they started a project plan. The CASA design and the modules were designed for reviewing DMFs, ANDAs, and NDAs, and they designed a IT platform which consists of all the review tools, and FDA is
kindly share
planning
your
to
screen.
implement this. Can you see? Can you see the screen now?
Sir, not it. Sir.
Can you see it?
Sir, yes.
Okay. So in 2021, CASA for the drug substance also applicable to the DMF reviews, and some sections of the ANDAs and NDAs. FDA has created IT platform which populates all the statistical data from these applications, and then it enables FDA to statistically evaluate all the data submitted in these applications. So FDA's plan is to implement CASA in future for review of the live applications, and based on that, the total data, whatever the analytical data submitted in these applications will also be populated into the IT platform, and they can review the data for approval process. Okay, so with this, I complete my at all. Any questions are there? I can interact with the participants, and I can answer any questions with respect to regulatory US regulatory questions. Yeah,
Sir, thank you very much.
Dr. Munir, I concluded my page. Thank you. And I don't know if at all anyone has any question on the regulatory affairs.
Sir, thank you very much, sir, for your presentation. Sir, there is one question here, sir, from a participant. Sir, the questionnaire was, what is ethical drugs?
Yes.
Sir, the questionnaire was, what is ethical drugs? Sir? that was a questionnaire from a participant. My name Dinesh Babu.
Dinesh Babu. Okay.
Yeah, sir.
So, yeah, I mean, in the generic market, when we are dealing with the FD, right?
Explain, yes. Disconnect type by
జాయిన్ అవుతున్నాడు మళ్ళీ జాయిన్ అవుతుంటే అవుతాడా Yeah, Dr. Puneet, as per my knowledge, there is what called ethical drugs. Ethical drugs is the prescriptional drugs. There is the drugs which can be prescribed by the doctors can be considered as the ethical drugs. Sir, thank you for the presentation of Ram Mohan Garu. And of course, he's in New York because of a midnight by 12:30. Maybe his net might be troubled there. That's why he couldn't come to live, and might be have some quality also somewhat deferred. So thank you for, sorry for the interruption, if any. Thank you, Dr. Ram Mohan, for your nice presentation topic on regulatory affairs on generic drugs. There is ANDS. Okay, thank you, sir.
Thank you very much, sir. Mohan, sir, very thankful for your addressing on regulatory affairs on NDA and ANDA. Now, I would like to take a privilege to invite speaker, Dr. Venkateshan Jayaprakash, sir, Associate Professor, Department of Pharmaceutical Sciences and Technology, BITS, Misra, Jharkhand, India. I would like to give a brief about Venkateshan, sir. He has obtained a diploma in pharmacy from Coimbatore and bachelor's of pharmacy from Madras Medical College in the year 1997 and master's program in the year 1999. And next, he has graduated research under the guidance of Professor Surendra Pandya in medicinal chemistry department. So subsequently joined JKK MMRF College of Pharmacy, Tamil Nadu in the year 1999 as an assistant professor. So later he has moved to Anjivani College of Pharmaceutical Sciences, Rajasthan. And in the year 2004, Sir has joined as joined in Department of Pharmaceutical Sciences and Technology, BITS, Misra, as a lecturer. Later obtained Doctor of Philosophy in the year 2011 under the supervision of Professor Haris Nayan Sinha on the theme anti-tubercular drug design. Currently, Sir is working as associate professor in BITS, Misra. And Sir has many collaborations. So he has collaborated with Professor Haris Nayan Sinha and Professor Rolf Hilgenfeld, University of Lübeck, Germany. Also with Professor Bruno Kennard, Maricel University, France, and Professor Velmurugan Devadasan, University of Madras, India. As a research group member in two Indo-German collaborative research projects on dengue and chikungunya, jointly funded by DBT, DMBF, and New Indigo RNF, respectively. He has more than 70 original publications in SCI Index journals. He also edited a book for CRC Trust in collaboration with Dr. Daniel Castagalungu, King's College London, UK, and Dr. Yusuf, Anadolu University, Turkey. And Sir has held many of the administrative responsibilities, such as member of Internal Quality Assurance, member Internal Complaints Committee, various departmental committees. Now I request Dr. Venkateshan Jayaprakashan sir to kindly deliver your talk, sir. Thank you, sir.
Thank you, Siva. Thank you for your brief introduction.
Yeah, first of all, let me thanks to Dr. Venkateshan for our kind acceptance in that heavy schedule. Hi, Venkateshan sir. Good morning.
Hi, Madhu. How I can deny? How I can?
Fine. Nice to meet you after a
Nice
long
to
time.
meet you. Yes, nice to meet you too. Okay, so I just would like to share few things because I am junior to your principal, sir, Madhuchivadan Sethi sir, Garu. Okay, so when I entered B Farm first year in Madras Medical College, sir was doing third year, and we were actually sharing the same floor in the hostel. He was just 2 rooms away from my room, room number 12. I remember that one still. Okay, and it's great thing after many years we could able to connect through this platform. Thank you for this opportunity, Madhu. Thank you very much. And okay, let me share my slide now. And I don't want to take much time, and I don't want to make things so technical. It will be a sort of general discussion. I think the audience are BPharm students, if I am right. Shiva, can you confirm the thing? May I know about my audience?
So your audio is very clear.
No, audience, whether they are BPharm students, diploma students, or MPharm students.
From all the streams, sir. There are research scholars also.
Okay.
BPharm, everyone out there.
Then that will be good enough. Right. So I can talk a bit regarding technical sheet things also. Share, shown, then, okay. Once the desktop, my desktop is visible on your end, please let me know. Okay.
Shades. Yes.
Okay. My slides are visible.
Yes, sir, it is getting shades.
Just a minute. Yes.
Yes, sir, we can see your presentation.
Okay. Are you getting my audio?
Oh, yes, sir. Yes, sir, we can
Okay,
see, sir.
thank you. No, are you getting my voice?
Yes, sir, your voice is clear, sir.
Okay, thank you. Okay, today I am going to share a few things. The title seems to be structure-based drug design, and under which I am going to talk a bit about molecular docking, right? So I'll be initiating my lecture from virtual screening perspective. Of course, yesterday Madhu has clearly told regarding the scenario, drug discovery scenario. And I'm going to touch the same thing in first few slides. And after that, one of our keynote lecture speaker also given few vital informations that are quite important when you are considering drug discovery pipeline. Okay. So the first few slides seems to be a repetitive information that you have already heard during yesterday lecture. Okay. Then slightly after that, I will move on to discussing some protocol regarding virtual screening. After that, I'll be touching upon what is molecular docking, and I won't be going in detail into molecular docking. Rather, I will tell you, if you want to practice molecular docking for the purpose of your research, then most of the time people do not know where to start with. So I'm going to give some information regarding what you need in order to initiate the practice of doing molecular docking or computer aided drug design, molecular modeling, whatever you say, right? So what we need before starting the thing, and how we should prepare ourselves to practice that one. So few information regarding that one. Then I will move on to a segment dedicated towards AutoDoc, because the one that is freely available and we are regularly using the tool. I'll be giving bit of information regarding AutoDoc. And after that, I'll be showing few information slides that tells how AutoDoc helped us in coming out with a good publications, right? So this seems to be the overall structure of the presentation today. And with this background, let me start with my first slide. And this seems to be quite, I think I'm getting the echo. Somebody kept that speaker on. Okay, right. So what you are seeing in the slide is actually a pictorial depiction of drug discovery pipeline that's being regularly practiced in industrial setup. Okay. And if you see the thing, yes, it is already been told that it takes more than 12 to 15 years. You can see the pictorial depiction, how much time is actually getting involved in each and every step in your drug discovery pipeline. And what you are seeing, a pie chart just below the flow chart, is actually giving you an information regarding the quantum of money invested in bringing one drug to the market. Okay, but the information that you are seeing just above the pie chart is the one collected in the year 2004, right? But the recent statistics says for bringing one drug into market, a company has to invest 2.6 billion. It's not a small amount. Okay, sometimes it seems to be more than the budget of a small country. So it's a huge amount that you need to invest in order to bring one drug into the market, right? But if you look at the pie chart, it gives some other information actually, right? So if you see, the blue region is actually saying effective cost in bringing one drug into the market. That's actually stands at 30% only. Means in 2.6 billion US dollar that you are spending, only 30% of that amount is actually spent on bringing one molecule to the market. So what about the 70%? Those 70% seems to be called as failure cost. Failure cost means a pipeline actually starts with multiple candidates, and from the multiple candidates, only one could able to come out as a drug in the market, and whatever the other candidates that were pumped into the pipeline seems to be failing at different stages in your clinical trials. So different stages of clinical trials, you can see the flow chart just above the pie chart. So in any one of the phase, those candidates are getting failed, and finally one could able to come out, and that one is actually consuming only 30% of this 2.6 billion US dollars, and other 70% seems to be consumed by other candidates that are failing at a different stages in your drug discovery pipeline. See, this seems to be the story behind why we require some computational tools. Okay, so every company is actually trying to find out a way thereby that they can reduce the failure cost, and thereby they need to make the business profitable one hand, and another hand they need to pump more drugs in the pipeline that are probable drugs rather than drugs that can fail at a different stage. Okay, so there comes your drug discovery. Means the early stage of drug discovery pipeline is actually starts with design and checking the hypothesis, synthesizing the candidate and checking the hypothesis whether it is actually giving the intended activity or not. Okay, so there we are using some computer aided designs, protocols, tools. Okay, and that actually seems to be working well, and quite efficiently companies could able to cut the failure cost. Okay, so we will see how they were actually operating in a subsequent slides, right? So this information has already provided by Madhu during the opening talk. For every 10,000 drugs that is entering into the drug discovery pipeline, means clinical trial setting, only one could able to come out as a drug. That's the thing I told you in the earlier slide. Multiple candidates will be entering the clinical trials, and one has the ability to enter into the market as a drug, and all the others are failing at a different stages. The slide is actually a pictorial depiction of the process where 10,000 drugs are entering into the pipeline, and one is emerging as a drug, and all others are failing at the different stages. You can see the thing. It's a simple pictorial depiction of what we are actually saying, right? And when you look at the thing, if company wish to reduce the failure cost, they need to understand why the candidate is failing. So unless otherwise, if I don't have an idea regarding why the molecule is failing at certain stages in the clinical trial, I cannot make any decision or make any mode to reduce the failure cost. So I should understand why these candidates are failing at a different stage in the clinical trials. And most of the time, if you look at the thing, the first pie chart on the left hand side top, if you look at the thing closely, you can see a large black chunk on the pie chart, pharmacokinetics 39%. Okay, it is saying that most of the drug candidates are actually failing due to their poor pharmacokinetic properties. Means if I could able to identify those candidates that may be failing in later stages in the clinical trials due to poor PK profile. Okay, if I can, I have a means to identify those candidates at the start of my drug discovery program itself. I can remove them at the start itself, and I can make most probable drugs to enter into the clinical trials. Means the companies actually need some predictive models. They need some predictive model in order to identify the drug that has the highest probability to bind with my target protein. When I say target protein, that are the enzymes or otherwise receptors with which I wish my molecule to bind with and show the activity. Okay, so I'll be terming that one as a target proteins. So if I have a means to identify or predict the most probable candidates that has the highest affinity towards my target protein beforehand, and if I have the model to predict the compounds that has the wonderful PK profile, a good enough big PK profile, and I can combine these two, means I can pick those molecules that has the affinity, best affinity towards my target protein, and also has a favorable pharmacokinetic properties, then I am ensuring that the molecule may not fail in the clinical trial process. Okay, and I can pump more such candidates in the pipeline, and that will increase the probability of getting a drug, and thereby I'm trying to reduce my failure cost. The molecule may not fail, or the failure seems to be much lesser than the process where I am not using these predictive models, I can say, right? So that's the idea behind bringing these computational tools in to the drug discovery protocol and trying to apply those things so that I can pick the right candidate at the beginning itself, before synthesis. Okay, I don't want to spend more time or more manpower or more chemical reagents in synthesizing n number of compounds. That seems to be the protocol earlier, before these computational tools were not available. We will be going on enumerating the chemical library and uh, putting lot of money, lot of manpower, lot of chemical reagents, and I come out with a library of thousands of molecules, and getting those things tested in animal models seems to be another headache, because CPC SEA is actually restricting the use of animals disease models. You cannot play irrationally. That's the thing. So you need to be rational so that things will fall in line, right? So there comes these computational tools, and I need to use these computational tools rationally to increase the probability of getting a drug from a pool of candidates that are entering into the clinical trials. Okay, so that seems to be the idea, right? Let's move on to the further discussion. Now the question comes, why new drug is required? Our first keynote lecture speaker actually talked about this one. Why we need new drugs? He provided many different reasons why we require new drug, and I'm not going to say something new other than the thing told by our keynote speaker yesterday, right? Still, I would like to highlight few things, and in case of antimicrobial agents, that seems to be the reason. Development of resistance that makes the molecule seems to be worthless after a period of time. Then we will be in a position where we require some newer agents to treat the disease. So this is quite common in antimicrobial segment, but you can see a similar trend is actually happening in many other pharmacological segment also. You might have heard about drug resistant or treatment resistant your depression. Okay, TRD they used to call, means even this can happen in pharmaceutical agents or pharmacological agents also. Resistance does not actually now stick on to or otherwise refers to only the antimicrobial or otherwise chemotherapeutic agents. Even pharmacological agents, and if you are using using the agent over a period of time, that will tend to show a sort of resistance or otherwise loss of efficacy, right? Earlier, that seems to be working at a dose level of 50 milligram. Gradually, you need to increase the dose to achieve the same pharmacological outcome. So it is means of losing its efficacy over a period of time. In all those cases, we need a new drug to replace the older one, ineffective one. And yes, of course, adverse effects. Adverse effects is the another thing. When a new drug comes into the market and when it was put in practice for over a period of time, after some point of time, when you start collecting the informations, you could able to know, yes, this drug is causing whole lot of adverse effects, and that's the point you will think of developing a new drug to overcome those side effects. So these are all few reasons why we require actually new drug. But and if you are looking at the another aspect, the major point or otherwise aspect that drives your drug discovery programs in the company is market. So what I mean to say market is nothing but you think a company is actually having a workforce, and establishments in different countries, and they have a exclusive marketing rights for few drugs, right? And those few drugs are actually generating a, quantum of money that could be able to support the existence of their units in different countries and support the workforce that is working in those units. Okay. And once the drug loses its exclusivity, then everybody starts preparing the drug or starts making the business with that particular molecule. Then the profit of the company will get reduced, and with that reduced profit, a company may not sustain its existence. So you need to understand this point. That means the company has to come out with a new drug regularly so that they can support their existence in the market. So that seems to be the major driving force why companies are investing more and more in drug discovery pipeline, and they will get it back in multiple magnitude, and that will sustain their drug discovery program in turn, and it will go on. It's a cycle, it will go on. Okay, right. So if that is the case, this drug discovery pipeline seems to be quite beneficial to the company's existence. Whether it is really helping the humankind society, if you see the thing, it's not going to help the society to a larger extent, because all the companies are actually investing on the segment where they could be able to get more return. Okay, so return on investment in terms of economy, they used to say, and they will be investing on those category of drugs where they could be able to generate more profit, and they won't concentrate or otherwise invest on a pharmacological segment or chemotherapeutic segment where the return of investment seems to be very less. So that's the thing. If you remember yesterday's lecture, where our keynote speaker said more than 75,000 of diseases are there, and only 25% of diseases we have the drug, and another 75% of diseases we have not developed any drug at all. Why? There are big pharma majors all around the world, but they are not showing any interest in developing any pharmacological agents or chemotherapeutic agents to treat those drugs, means diseases. The reason is they may not get the profit, and that's the region where academic research come into focus, play a role there. So if you want to do do the research in the neglected segment. If I said neglected, that is actually with reference to the industry. The industries are not interested to invest their money in that segment, and it is neglected by the industry, and academics research should be oriented towards the neglected disease. You put your research efforts to bring out come out with drug molecules to treat such a neglected diseases. So that seems to be the way the academic research can play a major role in the segment. So for that also we can use all these tools, right? So whether you are in the industrial research segment or otherwise academic research segment, these tools are quite helpful in minimizing your efforts in identifying a probable candidate that may be getting that may be hitting the market within a shorter period of time. So that's the thing. So you need to know, we academician also need to know about these tools, availability of these tools, and how it can be employed rationally to find a probable candidate in the neglected disease segment, right? Okay, so moving on to the next slide, the strategy. What strategy actually we need to adopt? We now actually encompasses both industry as well as institute, academic. Okay, so the goal for both the segment, both the institutions seems to be same. Come out with a new drug. They are playing in the profitable region, and academics should play in other regions, but they are also quite important. Okay, right. So how we need to formulate what should be our strategy so that we can come out. And this slide actually uh, uh, gives few points with reference to industry, but it is quite applicable to academia also, because uh, we just want to reduce the failure cost. That's common for both, whether it is industry or otherwise academic research. Try to reduce the failure cost. Then your fund is actually pumped rationally to probable candidates, on probable candidates. Okay, right. So when I say strategy, we need to concentrate on these points, the things that are appearing on the slide. I think it's quite faint, but still it is visible. I think so. So find some new targets. Don't work on existing targets. This is quite specific to chemotherapeutic agents, where the resistance seems to be quite common. And if you are working on the earlier existing targets, the microbes have already learned how to develop resistance towards any agent targeting that particular protein. So your new drug may not stay in the market for more time, because companies are very much worried about the life cycle. Not only companies, even any academic researcher, we want any new drug that is getting developed should stay in the market for more time to benefit the society, isn't it? And if the drug is quickly losing its lifetime due to the development of resistance, it is actually the effort that we are putting to develop a new drug is going vain. So try to make put your effort in such a way, any new drug that I am developing that's going to stay in the market for a period of time, right? Ensuring its life cycle, longer life cycle. Okay. So for that, you need to come out with a new target, identify a new target, and work on that, so that any novel drug that you are developing against the new targets will be staying in the market for longer time, because the microbe has to learn how to handle the novel drug targeted against a new protein. So the drug will stay more time. And moreover, if you are combining the new novel drugs with the existing drugs, you can make the drug to stay in the market for much longer time. So you might have seen the combination therapy that is actually being practiced in almost many different microbial infections. Okay, right. And this actually increases life cycle. That's the thing I am actually saying. So you will get a novel drug, and once you are getting a novel drug, with reference to industrial scenario, they are getting actually 12 to 15 years of exclusive marketing rights, right? And just one drug is enough for them to get the moreover, they will be earning the quantum of money. With that, they can sustain multiple of drug discovery pipelines that are running in their company. Okay, so that's the thing. So the idea is to increase the life cycle, identify the novel drug that works on novel target. Thereby, I am actually increasing the life cycle. And once I am increasing the life cycle, I can get the exclusive marketing right, and I can get my return on investment, and I can sustain multiple drug discovery pipelines in my company that will in turn give the next drug ready before this drug is losing its exclusivity, right? That's the idea actually, right? This is with reference to industry, but for academics also the thing is true, but we are not actually taking into the consideration the point, the profit. Okay, right. Now moving on to the next slide. It is actually giving the same information what I am actually talking, right? So the thing is, it is decreasing the investment and increasing the return. Okay. So now I am coming to the discussion on what positive points right now we have, because these things were not in existence if you consider a period 20 years before, right? So when there seems to be very less biotechnological development, computer architecture development, and Yes, these two segments or these two sciences actually accelerated your drug discovery program. One side we have biotechnological development, and on other side a computer science development. So these two emergence and the development of these two sciences or engineering branches actually accelerated the drug discovery program, what we are practicing these days, right? And if you look at the bar diagram that is given in this chart, it starts with 1976, and actually the three-dimensional crystal structure of a protein, people try to solve it and they started submitting those things in a database. We used to call it as protein data bank, right? And you can see the rise in the bar over the years, and now you can see more than 1 lakh protein. For more than 1 lakh protein, you have a X-ray crystallographic three-dimensional experimentally determined structure available at your disposal. And that is one factor accelerating your structure-based drug discovery. When I say structure-based drug discovery, for that you need to know the three-dimensional structure of a target protein, so that you can design your drug based upon the structure of the protein. Because I want my molecule to interact with the protein, and if I know the structure of the protein, I can design my molecule rationally so that it can interact with my intended target protein. That's the idea. So for that, I need structure of protein, and you can see the rising number of structures being deposited in your protein data bank. So you can find literally for almost all the proteins that are expressed, purified, identified, expressed, purified these days, you could able to get the crystallographic structure in the protein data bank. That's one positive point. If you want to practice structure-based drug discovery, the structure of intended protein, your intended protein, sure, I'm sure you could able to find the one in protein data bank. That's one positive point. And then the second positive point, if you look at the graph here, it is actually showing the data on publication with virtual screening or otherwise structure-based drug design or computational chemistry use of these tools in identifying a probable candidate, right? And here also you can see there is a consistent rise, means more and more publications started appearing. I think after 1990, you can see the thing, more and more papers started appearing. They actually utilized some or the other computational tools to design their drug molecules. That's the meaning. Okay, so virtual screening is actually being the norms these days to design and uh, uh, synthesize the compounds that may be a probable binders to the intended target protein. So you can see the thing, it's consistently rising, means the acceptability of the virtual screening seems to be rising over the year. And nowadays, if you want to be, if you wish to publish any paper in a high impact journal, they actually call for, even if you are providing the experimental data, they are asking you to support it with a simulation data showing the molecular level interactions. Okay, so that seems to be the thing. If you want to go for Journal of Medicinal Chemistry publication in JMC, ACS journals, or otherwise Elsevier journals, they actually ask for these simulation data. Let me show. Yes, your experimental data is there. Let me show how your molecule is interacting with your intended target protein at a molecular level. So without that, you cannot do the thing. That is actually one thing helps in further refining your design. So they want those things to be presented to the readers, right? So in both the way, in the designing and refining, okay, and in case of publication perspective, in every sense, now it seems to be mandatory that you start your design aspect with some computational tools and proceed further and you can claim that I am progressing rationally. Every step is rational, and I am not doing something haphazardly. Rather, I am driving my drug discovery through a rational approach. So that's the thing now everybody would like to see, right? Okay. Now, let me show the virtual screening protocol. The slide is actually showing a virtual screening protocol. So, in which there exist multiple steps. The actual thing what I am going to talk about in subsequent slides is receptor docking. So, receptor docking is the one, otherwise I can call it as structure-based drug discovery, right? So, that's the thing. Before coming to this stage, right? So, what you need to do, and after doing this, what I should do, okay? So, this actually shows a flow chart how one should proceed to approach a virtual screening for their research, right? Okay. So there is no need that every time you need to design with your imagination a novel drug or otherwise novel molecule. Okay. The reason is already n number of compounds, organic compounds were synthesized for some or the other reasons, and the structure of all those compounds are available either in publicly available database like zinc or otherwise NCI diversity data set, or something are actually with some commercial vendors. But zinc database is huge, more than 3 crore molecules are there. So there is no need for me to sit and imagine something and come out with a new drug, means new novel structure, right? And even I can do the thing, but the question is whether it is synthesizable. So all these things comes into picture. So just I want to use the thing, means there exists a huge chemical library that were already synthesized for some or the other reasons. I would like to use that one. I just want to know whether any of that particular any compound from that particular library is interacting with my intended target protein or not. So that's the idea. So that's why I am calling it as virtual screening. There exists a database, ligand database, and I'm going to use that one instead of simply putting my head hammering to come out with a new structure. Let's look at the library first. We will try to find any binders from the library, and after that there is a process that is called as lead optimization. There I need to make a rational modification that I will do, right? Okay. So, and now I'm going to consider that library, for example, which is having more than crores of molecules in it. But it is compute intensive. Actually, if I would like to screen all the crore molecules available in the database, I need intense computational resource. Even if you have an intense computational resource, it will take years together to complete the job, complete the task. Okay, right? If that is the case, the drug discovery pipeline is all about reducing the time and coming out with a new drug as quickly as possible, so that a company can get the exclusivity for a particular drug in a particular segment before any competitors come out with the thing and take the market in their hand, right? So they don't want the thing to happen. They want everything to happen within a short time span, right? So if that is the case, I don't want to put all these crore molecules in a virtual screening. Rather, I would like to rationally refine it first. So that's the thing I used to call it as database preparation. I'm not going to subject all the drugs in the database, means zinc database. Rather, I'm going to prepare my subset. The subset is nothing but, so there is no guarantee, isn't it? All the molecules will have a favorable PK profile, because most of the drugs, most of the chemical entities actually getting failed due to their poor pharmacokinetic profile in the later stages in your clinical trials. So I would like to predict that one at this stage. Okay, at the time when I start preparing my database, I will consider predicting which compounds in the database will have the favorable pharmacokinetic properties, so that I will consider only those molecules for my receptor docking, not all the thing. Okay, so there comes what is called as ADME admit filters. Okay, we call it as admit filters. It's nothing but a predictive model, and that will actually screen your molecule for favorable pharmacokinetic properties, and thereby the huge database is actually reduced to a small subset. Okay, so only the molecules that are having a probable pharmacokinetic properties profile as predicted by the model that I am having in my hand. So I am preparing a small database or a small subset. Okay, so after getting this, even the subset seems to be huge. If it is having 10 lakh, 20 lakh compounds, then even that is also taking huge time. I don't want to subject all these things, isn't it? So 10 lakh molecules also takes more time. So I just want to further pre-filter my compounds database on the basis of either similarity search or otherwise pharmacophore filtering, I can call, right? Means for the intended target protein of mine, the literature has already reported few compounds as a binders, right? And I will be taking those compounds as a reference, and with that reference molecule, I will go for filtering, pre-filtering through what is called as a similarity search or otherwise a pharmacophore based filtering. Okay, so that will further reduce my database. Okay, from the database subset, I am pre-filtering it to get a molecules that has a higher probability to bind with my targeted protein, because I got those features from already reported compound, and I am trying to see whether these features are available in the data set, and if such a compound is available, then allow them to move to the others will be neglected, rejected. Okay, so further pre-filtering is actually reducing my database to a very smaller one having few thousands of compounds. Okay, so now I am in a comfortable position because I have very few thousands of molecules, and I can go for an intense docking program, and that will return me a much probable candidates after doing a receptor docking. And I'll be picking top 10 or otherwise top 20 after doing my molecular docking, and those 20 compounds will be either procured from the vendor or other case they can be synthesized in the laboratory. You see, I have reduced the job of synthesizing multiple compounds, enumerating multiple compounds. Rather, I am picking the 20 probable candidates that can be either purchasable or otherwise for that the synthetic protocol is already available. I can do it in my lab. I can synthesize those compounds, and those things will be subjected to experimental protocol. Okay, so I just want to see whether experimentally it is really inhibiting the enzyme or otherwise not by means of an in vitro enzyme inhibition assay, and that will give me a lead, and that lead will be further optimized to get a drug candidate, and that takes a whole lot of procedures. But I am not going into that segment. Rather, I am trying to stop my discussion up to identifying the probable hits, the top 20 hits. How to identify the top 20 hits? Definitely, the top 20 will give me one or two probable hitters, right? So that's the idea behind doing this virtual screening protocol, right? So by saying that one, now I am moving on to receptor docking. What it actually means? Structure-based drug design is all about receptor docking. So let's move on to a discussion on molecular docking, right? So in a simple word, I would like to explain what actually docking means, because you know the thing, we all studied biochemistry and we know the lock and key theory behind the enzyme substrate interaction, right? So I would like to explain the same with the same analogy. I have a log that's nothing but my target protein, but I don't have the key with me. Then I would like to find one key that could able to open the lock. So I have a bunch of keys. Okay. So this is nothing but the compound library that I am talking about, zinc database, right? and I will be doing a process in which I will check with each key whether it is opening or not. That's the thing I am doing, molecular docking here. I am bringing those two molecules together and see whether they are interacting properly or not. So that's the thing I am trying to do with each molecule in the library. Then at one point of time, I could able to find out the key. So means molecular docking is nothing but finding the right key for the lock, nothing but finding the right compound for the intended target protein. Okay, right. So saying this, now the question is what I require, means if I would like to practice molecular docking, so where I should start with. So what I need for practicing my molecular docking, that's the question right now. Okay, so many B.Pharm students are there, M.Pharm students are there, even PhD scholars are there. So they wish to practice the thing for their research, and they do not know where to start. And most of the time, if some commercial softwares are available with your institute, the thing seems to be quite easy, and those things are clicks away, right? I can say clicks away. There are few windows in the drop down menu, and you need to click, 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 and it will give you the result. But in case if the institute is not having any such commercial softwares for the purpose of doing or practicing molecular docking, the option is open source softwares. Okay, so don't worry about the availability of software in the institute, and if you could able to learn how to do the things with the open source tools, then you are independent. You are independent means you could able to download and install it in your system, because nowadays everybody is having their own system. After this pandemic, you can download and install those tools and start practicing it. That's the good thing. Even after you are moving out of the institute, it is always staying with you, and at any point of time you can practice the thing, right? So what I need to have in order to practice the molecular docking, that's the question, and I would like to answer these things. I want structure of intended target protein, obviously, right? And that I can get from some sources. I would like to tell all those sources in subsequent animated material. Then I need a structure of ligand, right? So that may be either I designed a library on my own, considering the literatures existing earlier already, or I can depend on the database that are available already publicly in public domain. Okay, then I need a software program to bring these two things. Okay, my protein and my ligand. So bring those two, and let us try to see whether they are interacting or not. To perform that one, I need a software, right? So I need these three things. I want to see whether my intended target protein structure is available, and if my I can able to get some publicly available ligands or not. Then I need to see whether I can get some softwares available open source. Okay, so I can do it. I can do it without depending on anyone, right? If these things are available, let us see what are all the options that are at our disposal, right? So for structure of intended target protein, so where I will get the thing. So I told you that structure of intended target protein, you can search the availability of three-dimensional structure of your intended target protein in protein data bank. The website address is given. Then if you are not having the structure in the protein data bank, then you can depend on what is called as homology modeling, right? And even if homology modeling homology models are not available, and if you don't know how to practice homology modeling, then nowadays Google has come out with AlphaFold that's much more precise than the homology modeling that we practiced earlier, right? So you could able to get, means either you will be getting a experimentally determined three-dimensional structure of the protein in the protein data bank. If it is not available, nothing to worry. You can model your protein based upon the homologous proteins available in your protein data bank, either using a modeler or otherwise using AlphaFold, right? you can get. So don't lose hope when you can't find any structure for your intended protein in protein data bank. You can model it, right? Even the modeled proteins are working well, and many such literature references actually assures you. Okay, right. Then the question is, where from I can get the structure of my ligand? So either you can design your own library, or otherwise you can depend on publicly available library, the one that I told you, Zinc and NCI, right? So these are all the sources of my ligand. And then when it comes to the software program, more than 60 software programs are available for performing your molecular docking. And quite famous and probable highly cited software seems to be the one I am talking about in the next coming slides, right? Right. So I'm trying to give some examples for commercial softwares. You have a Gold, Flex, ICM, excuse me, Glide. Okay. So apart from these, you have n number of tools, but I have listed few things that are having good citations, right? And more citation I can say, right? On the other hand, I am trying to list out academic free, means you need to register yourself in their site and they will be availing the tool free of cost for 1 year period or otherwise 2 year period, and it is renewable, that you can do. Or otherwise open source means without any registration you can download simply from their site and you can start using it, right? And I have a list of few such tools here. Autodoc, Doc seems to be quite free for academic community. So you need to fill up a registration form to download the thing. You can do it quite easily. And Fred, actually they are offering 1 year license for academicians, and you can also get it from OpenAI tools. And these are all academic free. And why I am listing these things means that they have more citations, right? And there are open source tools. They gained very few citations, means scientific community acceptance seems to be quite low. But when you see the name of these tools being used in your research, and when you go for publication, it seems to be highly accepted tools actually, right? Now moving on to why Autodoc, because Autodoc we are regularly using in our research, right? And the question is, why Autodoc? Autodoc is actually open source one, I can say academic free one. And if you see, they started issuing this tool free of cost to academics, right? Their commercial price, I mean non-profit organization price is quite different. It is free of cost for academicians since 1990. And if you see, till Autodoc version 3, it seems to be running on command-based mode, right? And mostly the people working in the area of protein-protein interaction, biochemists, biotechnologists, okay, and sometimes chemists were actually widely using this tool, except pharmacists actually, right? Medicinal chemists. We started using these things much later. Once Autodoc 4 appeared, Autodoc 4 actually incorporated more atom types, including atom types for the small molecules, and that's the one actually designed for performing a protein small molecule interaction studies, right? So since then, the Autodoc seems to be quite popular in medicinal chemistry segment, and people started using this tool for designing their drugs, molecular modeling aspects. Okay, so that's the thing. And moreover, Autodoc 4 is actually comes with graphical user interface. They used to call it as molecular graphics laboratory tools. MGL tools is their GUI, and they have drop-down menus where you can prepare your protein, prepare your ligand, and prepare your docking protocol and run your docking. Everything you can do it in your graphical user interface, and it seems to be quite easy for medicinal chemists to adopt. Okay, so since Autodoc 4 emerged, MGL tools started appearing in the market. Okay, it seems to be quite preferable tool. So if I say simply that one, you may not be getting convinced. So I am putting one more slide here. And this seems to be a study conducted by a scientific group in the year 2006. And even in 2011, you can find a similar sort of statistics in the Autodoc forum itself. And in both the cases, in the pie chart, you can see the number of citations per few tools. They have actually listed 25 docking tools, and the citation seems to be much more for Autodoc in everything. You can see the thing, the blue chunk in the right top and on the black chunk on the right bottom, okay, pie chart. You can see the thing, Autodoc is still highly cited software, means highly accepted by the scientific community, and it is free of charge, right? So you can use it for your research. And moreover, there are some success stories. You need to know those things also. Then only you could able to get yourself convinced, isn't it? So you know the thing, the integrase inhibitor, right? Raltegravir seems to be the one developed by Merck, and they got a FDA approval in 2007. And the molecule was actually developed by a group of scientists under the leadership of Professor J. Andrew and James, actually. But the initial molecular modeling aspects related with this compound was performed by Joseph E. Mayer. He is actually chair of theoretical chemistry at University of California, San Diego. Okay, and from there they picked up and they developed that one into a drug, and now this drug is available in the market. So it's one success story of your Autodoc, and many such cases are there, but yes, those things are not actually publicly declared. Okay, so this seems to be one good example about the success story of your Autodoc, right? And moreover, the Autodoc is actually a preferred tool in many parallelly running computational resources where you can run your virtual screening. If you wish to submit your job, submit your library, designed library for a neglected disease segment, all these grids are actually meant for identifying a drug for a neglected disease. You can see the thing, dengue, malaria, okay, then childhood cancer, influenza. So there are many different computational grids that are available where you can run your virtual screening. They made Autodoc as a core program for the virtual screening, and n number of examples are there. Means it is actually showing the wide acceptance of your Autodoc for the purpose, right? Okay. So now I'm going to discuss about what's our experience, and with a few examples, I would like to conclude this lecture. So in 2006 and 2007, I was actually doing my pursuing my PhD in Birla Institute of Technology, and my research title is anti-tubercular drug design for the treatment of tuberculosis. And as I was talking about novel targets, my target seems to be the one less explored, and for that no inhibitor seems to be reported elsewhere, right? So I was actually working on the mycobacterium pathway, and the time when I was working during 2006 to 8, if you see, I could not able to find any crystal structure for my intended target protein. My intended target protein seems to be phenyloxazoline synthetase in the mycobacterium biosynthetic pathway in mycobacterium tuberculosis, and it was not available. Even I was unable to find a homologous protein structure in the protein data bank, so that I was literally not in a position to practice my molecular docking in order to say my design seems to be rational approach. Rather, I went on to say my design is rational based upon the substrate structure. So that was the thing earlier medicinal chemists used to do. Based upon substrate structure, they designed their inhibitors. Yes, of course, that's the rational approach, right? After these computational tools started appearing, the meaning of rational approach slightly deviated, right? They actually considered structure of protein rather than structure of the substrate. That's the only difference. Earlier, it seems to be the structure of the substrate for rational design. Now it seems to be the structure of protein for rational design. Okay, right. So during the same time, if you see the my design molecule, you can see the structure on the right hand side. I was actually designing a pyrazolene, and that is actually having the structural similarity with the Yersinibactin and Mycobactin. I'm trying to show the similarity in this particular pictorial depiction, right? You can see that these compounds are having similarity with a segment of Yersinibactin and a segment of Mycobactin, right? So these compounds, the library were actually screened for antimycobacterial activity as well as for Yersinia pestis also, and many compounds seems to be working well, and we got a good publication out of that. But at the same time, we came to know the pyrazolene seems to be working well for depression also. Okay, and they there seems to be a probable candidates or otherwise uh, a good scaffold for designing a monoamine oxidase inhibitor, MAO inhibitors, and they'll be finding their utility in the treatment of depression, right? So parallelly, we initiated another program. Of course, the main idea is to identify the antitubercular drug, and at the same time, we parallelly initiated, because the scaffold seems to be pyrazolene working well for monoamine oxidase inhibition, we initiated that one also for the same set of compounds, right? So once we shifted, once we started parallelly running that one, that time we got a option to practice molecular docking. Okay, because the structure of human monoamine oxidase enzyme was available, still available in protein dentagram, right? So that gave me an option to rationally say, yes, these compounds are actually binding with my intended target protein. So that point of time, in order to say for this one, no problem. Of course, for pyrazolene, we have a literature reports that they are actually working as an antidepressants, but still we would like to show the molecular interaction, right? So the problem with our hand at that point of time is which software to use to run a molecular docking program, and on which operating system, right? So we were actually having Microsoft based systems, and at that point of time, your Autodoc or MGL tools were not available for Windows system. It is available for Linux system, and we were unable to handle Linux system at that point of time. Even today, if you see, if you ask many of us, many of the chemists and otherwise, many of the medicinal chemists, pharmacists, they are quite comfortable with using Windows system. When you say the system is Linux, then they will bit shy away from the thing, right? So that was the case, and during 2007 and 8, all the systems available in our institute seems to be Windows based, and we were not having the Linux based system at all. And we are not courageous enough to install Linux and run the program also, because we were very much unfamiliar with Linux system, and we don't want to learn also. That's the problem with the medicinal chemists and pharmacists also. Okay, so we don't want to learn the thing, and we just want to have something easy at our disposal so that we can practice it. So you need to invest some time to learn the thing, and Linux systems are quite easy to learn and quite productive and quite fast in comparison with the Microsoft Windows, right? So we don't have the option at that point of time. We don't want to use the Linux system, but we used Linux emulator meant for Windows system. Means within Windows system, I can run a Linux terminal using Sigwin. The option is called Sigwin. We used that Sigwin program to run the Autodoc, but took longer time. The problem is I don't have a huge library. I was having a 50 compound library, so that was quite fine. Even if it is taking 2 to 3 hours to complete one docking, 50 runs, no issue at that point of time. We used that one, right? And we picked Autodoc, and with Autodoc, since it is open source, okay, nobody is around us to help. Means if you are stuck at any point of time, nobody is around to help you. Nobody is around to help you because nobody knows how to run it. So we were the first team to install Autodoc in our institute and start running it, right? And if something happens, and if the program stuck at any point of time, we are blank. So only rescue seems to be we registered ourselves in Autodoc forum and we start putting our problems in the Autodoc forum, and some people get respond, means they responded to our query and they availed some solution to our problem. They tried to explain why it is happening and how it has to be handled. So it took almost 8 months for us to practice and get an expertise in running the Autodoc, right? That point of time, myself and Murugeshan, who is right now actually in BITS Pilani, he was also doing a PhD along with me in my lab. So we were actually trying to learn and run the thing, and except we two, nobody else is around to help us, and we were regularly interacting with the forum to get the problem solved, and somehow we perfected it, right? But now the scenario is quite different. If you go and just enter Autodoc in YouTube, you can find n number of videos, and each and every video is clearly saying the way you need to start, the way you need to complete the the protocol. But at that point of time, 2007 and 2008-9, you cannot find any material except the material in Autodoc web page. There is no video, there is no protocol, there is no tutorial. Only the forum seems to be the available thing, and we utilize that one to perfect ourselves. So that seems to be the thing, right? And now we are quite comfortable in running it, and all our students are regularly using this one for their research, right? And I would like to show the relation why we wish to go for monoamine oxidase inhibitory activity. So you can see the similarity of my compound with isoniazide and iproniazide. Okay. So in both the cases, you can find the structural similarity existing in our compounds. Okay. So this aryl ring and hydrazine segment here. Okay. So you can see these things. So that's why we wish to go for monoamine oxidase inhibitory activity screening. And the first publication we got, and all our compound has shown very good monoamine oxidase inhibitory activity. Most of the time they are monoamine oxidase A inhibitors, right? And we got the activity data from one of our collaborator in Turkey. She is actually working in Hacettepe University. Now he is the head and dean in the university, in the biochemistry, School of Biochemistry. And we used to collaborate with her, and they provided the activity data. So we had the data with us. When we went for publication in Bioorganic Medicinal Chemistry letters, so we submitted only the activity data, but the editorial board and the reviewer started asking for provide some molecular level interaction through computational studies, even after submitting the experimental data. So that's the point we started learning molecular docking. So since then they started asking, even if you have experimental data, And if you are especially having the experimental data, one-to-one interaction with the enzyme, because all these things were evaluated on a enzyme assay, means my molecule is made to directly interact with the enzyme in a 96 well plate format, right? So in such case, they definitely require a support, molecular level interaction support through some computational tools. So they asked for it, and we are supposed to provide, and we learned, and we provided the data through molecular docking studies. So this was our first publication that came out in 2008, and we could able to show the molecular level interaction of potent compound with monoamine oxidase A as well as B, and the pictures are provided here. All these pictures were captured from the MGL tools that was also freely available from the Scripps University website. Okay, and the next is actually we started answering the questions of reviewer, and thereby we were actually trying to learn everything one after the other. So my sincere advice or suggestion you can take, if some article that you have submitted somewhere in some journal, if it is getting rejected uh, with few comments from the reviewers, don't try to resubmit submit the article to another journal. Rather, you work on the comments given by the reviewer, try to bridge the gap, then resubmit the same article to the journal after filling up the gaps, right? So this is the only way you can learn. Otherwise, most of the time people used to do, yes, if it is getting rejected in a high impact journal, let's move on to the low impact journal. So you are not going to learn anything. You try to bridge the thing. You try to answer the queries by doing additional work, and that's the only way you can learn, right? So this is my sincere suggestion. You can take it as a suggestion, and we were actually doing that. So first time we provided the data. We were actually evaluating our compounds using rat monoamine oxidase isolated from the rat brain, but we run our docking simulation using human monoamine oxidase. So reviewer are actually asked for how you are correlating. So it's quite you need to rationally answer the thing. You are performing the experiment in rat monoamine oxidase, and you are doing your simulation on human monoamine oxidase, and how you are correlating the thing, how it can be correlated, right? So then we need to do a sort of research, literature research, okay, survey, and we need to see what seems to be the sequence similarity, sequence identity between a rat and human. Then we need to come out with the data that the rat and human seems to be sharing a similarity. They are homologous, so it can be accepted. Sort of things you need to give in acceptable way. You need to provide your discussion, and that's how we started learning each and everything. So why I am putting this, when you are running a simulation, see that your enzyme system used for experiment and the one that you are using for simulation seems to be similar. Otherwise you will be getting a comment from the reviewer, and you need to answer it in a rational way, right? So we experienced all those things. That's why I'm putting the thing. Then we could able to get a project from AICTE, and we actually come out with a script. So at that point of time, nowadays Autodoc Vina seems to be quite popular for running a batch processing. I mean to say, batch processing means you can input 20 to 100 ligands at one click, and docking will run one after the other, and it will throw your result after some point of time. Okay, so that we used to call it as batch processing, right? But Autodoc is not actually having an option to run batch processing. Means you cannot submit 20 ligands at one go. Rather, you need to submit one ligand at a point of time, and once it is completed, then you need to make a second input. Okay, so batch processing was not available at that point of time, and we made our own Linux script to make it run in a batch processing mode. and we got that one published in a bioinformation journal. So remember that point of time, Autodoc Vina was not available, but we could be able to do the job through a Linux script. And this we did in collaboration with our computer science department, engineering department, and it was still working well, right? And we used this script in subsequent docking programs actually. So this is some screenshot that we are trying to show you the script that we developed for batch processing. Okay, then this one is actually trying to show the overlap of experimental data with the simulated data. You can see the thing. The graph is actually showing what we could be able to get through simulation is quite exactly matching with the experimental data that we generated with the enzyme inhibition assay. Don't expect that this will happen every time. Okay, so sometimes it may happen, and most of the time it may not happen, because we have a parallel experience with the histone deacetylase enzyme. Autodoc has predicted very good interaction, but when we went for enzyme interaction assay, all the molecules failed. Means Autodoc program is not suitable for screening histone deacetylase enzyme inhibitor. Remember this fact, not all the programs is suitable for all the intended target proteins. Okay, you need to find the one probable one by means of a validation. I am not going to talk about validation and all in this lecture. So in case if I have an opportunity in coming days, I would wish to take a workshop sort of thing for practicing Autodoc. Okay, so if time permits, that's later on. Okay, so Autodoc perfectly suited for monoamine oxidase, and we could able to get a very good correlation between experimental and the simulation data. So that's the one we presented in our next publication in 2010, right? And one more problem at that point of time seems to be the forum itself. Autodoc forum itself says Autodoc is unable to handle the enantiomers. Okay, and our compounds are actually having one chiral center, and there is a chance I have just few slides. I'll be concluding it. Okay, so just I would like to say the way that Autodoc actually handles the chirality also, right? So if you look at the molecule, the fifth position of the pyrozoline is actually a chiral one, means there is a possibility that I can have two enantiomers. When I am synthesizing, what I am getting seems to be a racemic mixture, but the reviewer started asking, you are presenting a result, simulation result, so for which isomer you are doing the simulation? Is it for S configuration or otherwise R configuration? And how you are going to correlate the result, experimental result, with these isomers? So that seems to be the next challenge we faced. And when you look at the Autodoc forum, they many of the people actually reported that it is unable to handle the chirality. But when we ran our isomers prepared, sketched using, we were actually using Mervin sketch for sketching our compounds. It actually provided a different scores for these isomers, and the chirality seems to be retained during the process of docking. Many people reported that R configuration is getting changed to S when I am running the docking program, and S is getting changed to R. And these sort of reports were there in the Autodoc forum. So we were a little bit confused whether to use the tool or not. But when we used the tool, and we could able to see there is no change in the configuration, it retained the configuration, and we could able to get the score for individual isomers, and we could able to show the correlation of these isomers with the experimental data, considering we have the racemic mixture. Okay, and we got one good publication on 2011 after answering these queries of the reviewer. See, we also learn one after the other when we start answering the reviewer's query. So that's why I'm suggesting keep working on reviewer's comment. Don't try to submit somewhere else. If you could able to answer the query, then you can resubmit there itself and you can gain a paper in a good impact journal. So that's how you need to work and learn, right? So this is regarding our experience. I don't want to extend beyond this. And of course, we were still in the process of learning how to perform ensemble docking and covalent docking that we will do in coming days. And with this, I think this is my concluding slide. And we are continuing our journey to learn covalent docking and considering the protein flexibility and ensemble docking through ensemble docking, and we'll be doing it, right? Okay, thanks for this opportunity. Thanks Madhu, thanks Shivashankar Reddy. Okay, and for all the attendees, those who are attentive throughout this session, and in case if you have any doubt, I'm here to answer it.
Thank you very much, sir. Sir, there is one of our questions. After docking predictions, shall we go synthesis directly without any references?
Okay. So after docking and when you are come out with a hit, right? And if it is the molecule designed by you, then you can directly go for synthesis. Once you are convinced yourself, because this you are doing, the molecular docking simulation that you are doing to convince yourself. First of all, I just want to convince myself the designed compound is actually binding with my intended receptor. I just want to confirm myself first. Then only I can convince others, isn't it? So I need to get convinced myself. Yes, this one is binding well with the target. So I can go for synthesizing the compound. Then you can go for it.
Thank you. Thank you, sir, for your excellent talk. You also enlisted about the drug discovery pipelines. How our efforts should be placed whenever when we are discussing a new, developing a new molecule in terms of a long time, ensuring a long time of a drug molecule, and also giving various enlisting of various softwares regarding docking studies. So this will help you help all the students, including with the scholars and faculty members, to utilize the online docking sources. And thank you very much, sir.
Welcome. You are most welcome.
One small question, sir.
Yes, please.
Now, Autodoc has become a paid version, sir. Is it right?
No, it is still free. It's not a paid one.
Sir,
only for industries, non-profit organization, they are having a price tag. And for academics, it is free. Only thing you need to fill up a registration form. That's it. So there is a question from Padmini Dudi Mani.
Yes, sir.
Yes. Yes, please go ahead.
Padmini madam, you are free to ask a questionnaire. You can clarify your.
I think she is unable to unmute herself.
Yes. Padmini madam, you can ask the question. You can clarify your doubt.
sir, how about molecular dynamics simulation, sir? When should we go for molecular dynamic simulation?
Okay. So really a good question actually, right? So molecular dynamic simulation is much more precise than molecular docking. Okay, but the problem is it takes much intense computational time. Means just for running a 10 nanosecond simulation in a high performance computer itself, it takes days together. So most of the time that's not suitable for a virtual screening approach. So we need to stick on to a protocol where I can get the result quickly. So that's why the docking comes first. And when you have a handful of hits through molecular docking, those hits can be subjected to molecular dynamics. So it comes next to your molecular docking. Okay, so I am subjecting thousands of ligands after pre-filtering, and that provided the hits in the ranked order. I can pick top 20 compounds, and those top 20 compounds can be subjected to molecular dynamics in order to get much more clear insight. I hope I cleared your doubt.
Yes, sir. Thank you, sir. Thank you very much.
Welcome.
Now, with the permission of principal sir and organizing secretaries, we would like to felicitate Dr. Venkateshan Jayaprakash sir virtually. So kindly accept our virtual felicitations.
Sure. Thank you.
Sir, I request you to stop your sharing, sir, so that we can share our felicitation slide.
Thank you very much. Thank you.
Thank you, Dr. Venkateshan.
Thank you, Madhu. Hope we.
Well planned lecture.
Thank you for the opportunity. Thank you.
Sir, thank you all the delegates. Sir. I request the delegates to join in the afternoon session at 1:30 p.m. There will be one more wonderful session with Dr. Yoganjanalu sir on Indian patenting. I request the delegates to participate in the afternoon session.
Okay. Thank you, Siva. Thank you, Madhu.
Thank you very much, sir. Correct.